All right, so video 1 talked about our linear or open chain carbohydrates, trying to get this in the most stable configuration, right? Zigzag. So we've got glucose in its open chain form, very reactive because we have a free carbonyl containing functional group, an aldehyde in the case of glucose. So very reactive. So what does nature do? Nature cyclizes it. Cyclization converts that aldehyde into a hemiacetal in the case of glucose or a hemiketal in the case of fructose. That protects that. But we're not as fully protected as we could be because we are one step away from having a free carbonyl. So cyclized sugars are considered reducing sugars. The next thing that we want to do is we want to double knot our shoelaces. That means we're going to take and react a second equivalent of alcohol with our hemiacetal or our hemiketal, and that's going to generate an acetal or a ketal. We've double knotted our shoelaces. That is a non-reducing sugar because that anomeric carbon's OH group or that anomeric carbon is tied up in what we call a glycosidic bond. Okay, so walking through the mechanism for what we see there. Okay, again, highlighting this essentially picks up where that fourth reaction mechanism left off. We had a hemiketal. So what we're going to do is we want to add a second equivalent of alcohol here, where we have an sp3 hybridized carbon atom that we need to substitute essentially one nucleophile for another. We don't want an OH group there. We want an OR group. Anytime you want to make something leave a group, protonate it. So we're going to protonate this OH group. We generate water that can easily leave when pushed out by the oxygen, the heteroatoms, that oxygen's lone pair, generating, again, a very electrophilic reactive intermediate here. Now we've got nucleophilic attack by another equivalent of alcohol. Okay, deprotonation of that incoming nucleophile generates our hemiketal. So mapping on some of the things that we already talked about when this chemistry has to do with sugars. Okay, so we already said, well, what if your incoming alcohol group was connected to the rest of your molecule that had your carbonyl containing functional group? That allows us to generate a ring that allowed us to cyclize our sugars, and we formed what we referred to as the anomeric bond. So that is the bond formed between the anomeric carbon, the carbon that was the carbonyl, and our incoming nucleophile, which in the cyclization of sugars is that C5OH group. So again, that is how we cyclize our sugars. Now what we're asking about is what can we have then as an additional nucleophile? And we just walked through the mechanism for how that happens, but we're going to have another nucleophile come in, okay? And we're going to generate then, by nucleophilic attack, we're going to generate a new bond, and this is a glycosidic bond. So I know I've said this multiple times already, but let's highlight. When we cyclize our sugars, we generate what we're referring to as an anomeric bond. That allows us to form hemiacetals or hemiketals. We've knotted our shoelaces. We've protected that carbonyl containing functional group. When we react that cyclized sugar with another nucleophile, we're going to talk about the fact that there's lots of different options. In this case, in our mechanism here, we see it as another alcohol group. What we do is we generate a new bond to that carbon, and we refer to that as a glycosidic bond. Okay, biochemical application for this, again, we are going to be cyclizing our sugars when we form hemiacetals and hemiketals, and then we polymerize them through the formation of a glycosidic bond. So we polymerize sugars. We're going to talk about glycosylating proteins, and we're going to talk about adding nitrogenous bases to sugars. So 3 applications that we can see for using this formation of a glycosidic bond, this chemistry. Okay, so again, let's talk about some specifics for sort of what we see here. Okay, and again, the key part here is asking what other nucleophilic possibilities do we have here? What other possible nucleophiles? So highlighting what we sort of see here with nucleophiles, and this is the case for what we see in all of our sort of biochemical reaction mechanisms, is nucleophiles tend to be heteroatoms like oxygen or nitrogen. So I'm going to split this page in half here because we're going to talk about essentially those other two atoms that we can have. We can have an oxygen as part of an OH group. Okay, we can see that coming from other sugars. That's right, other sugars here. Okay, and when we have other sugars, we are going to generate polymers. That's how we polymerize our sugars. Those, again, are polysaccharides. Okay, I'm going to pull out my sugar back here. Right, here's glucose in its cyclized form, right, showing it in the chair conformer, or if we look at it as a Hayworth projection, that oxygen is always going to be in the back right corner. Let's look at the different OH groups that we still have, right? I've got an OH on my anomeric carbon. That's why it's a reducing sugar. That's why it's reactive. It still has an OH group here. I want to point out that there's another group that's going to become important. We've got another OH group at our C6 position. That's going to have some application in just a little bit here, okay? But we've got other sugars that have OH groups that we can start polymerizing them together. So looking at sort of my hand gestures here, we've got linear sugars that we cyclize, and then we've got cyclized sugars that we can link together and polymerize. So I want to highlight here that we know that we can have OH groups from other things. We can have OH groups from amino acids. Specifically, we know that they can come from serine, threonine, and tyrosine amino acids. What's going to happen with this sort of chemistry is we're going to take proteins then and be able to hook sugars to those. We call this glycosylation. Just going to give myself a little bit more room here in the center here. This whole process is called glycosylation. So glycosylation, really referring to that glycose piece, really means sugar, or specifically glucose. And that ylation, okay, that means that we've added. So glycosylation is really the adding of sugars. So when we talk about that nucleophile for this chemistry, right, thinking about what nucleophiles do we have, that OH can come from other sugars, 100%, and that's going to generate polymers or polysaccharides, but we also can have that OH group come from 3 specific amino acids, serine, threonine, and tyrosine. That is how we are going to hook sugars and proteins together. We refer to that as the glycosylation of proteins. We're going to talk about some examples of that in just a second. The other piece, though, that I want to highlight is the other sort of atom that we can see that's a great nucleophile is nitrogen. Specifically here, we can see that there's amino acids that we have for that. And again, these are going to be our weird letters because they have functional groups that do chemistry. We've got arginine, we've got asparagine, we've got glutamine, we've got lysine, and we've got histidine, right? All other amino acids, we've got 8 different amino acids that we could use as anchor points to attach sugars to proteins, okay? The other place that we can see nitrogen atoms that could be nucleophiles are the nitrogenous bases, like we see with our nucleobases. Regardless, we can refer to these as either O-glycosylation if we have our nucleophile as an oxygen atom, or we've got N-glycosylation when our nucleophile is a nitrogen, okay? So highlighting places that we can see this, I'm going to zoom in here just so that we don't run out of space here. We can see this when we're generating nucleosides or nucleotides. Okay, remember nucleosides are a nucleobase attached to the sugar. That point of attachment is an N-glycosidic bond. Nucleotides just further modify a nucleoside by adding between 1 and 3 phosphates, right? So we've got that chemistry that we see here. I also want to bring up and highlight two examples that we're going to see as we move through the course here. We're going to see hemoglobin A1C, and then we're going to see our ABO blood groups. All right, hemoglobin A1C. So one of the giant threads that we have in this course is talking about glucose homeostasis, right? So understanding what your blood sugar is really important. One way that you can get your blood sugar is by just kind of using a glucometer and getting an instant reading, really a snapshot in time of what your blood sugar is. But that can fluctuate greatly. So oftentimes what medical professionals will use is something called a hemoglobin A1C level. So what this is you are actually looking at the glycosylation level of the protein hemoglobin that's in your red blood cells. Because red blood cells live on average for about 3 to 4 months, what this really represents is sort of a time-lapsed average of how sugary your blood is. The more sugary your blood is over that 3 to 4 month time period, the more glycosylated your hemoglobin will be in your red blood cells. So that number that you see for your hemoglobin A1C is really a percentage of your hemoglobin that is decorated or glycosylated with sugar. Okay? The higher your average blood glucose is over a 3-month period, the higher your A1C level is going to be. So we'll spend a lot more time talking about that later. The other thing that we see with this sort of glycosylation chemistry is the ABO blood groups. So when you talk about the antigens that are present on the surfaces of your blood cells, those antigens really are carbohydrates and different carbohydrates that are decorating your cells, whether you're type A, type B, or type O. And that has to do with not only the sugars that are on your cell surface, but the antigens and those are the antigens, the antibodies that you actually have in your blood that would recognize those blood sugars. So lots of neat applications that we have here for some additional chemistries. Okay, to sort of, I think I might stop there.